In the first part of 4.2, we discussed passive transport. In the second part, we're going to discuss active transport. And active transport is like what it sounds like. It's active and it requires energy. It also requires carrier proteins, and we've talked about those before, that are in the double lipid bilayer of the plasma membrane, and it's going to combine with molecules to get them across, going against the concentration gradient. And because it has to go against the concentration gradient, which we talked about, if you imagine going on a float trip and having to actually paddle upstream against the flow of the water, it's going to require extra energy, and that's going to be an active type of transport because you're moving against the concentration gradient. This can be entering or leaving a cell. Energy is always required. So let's take a look at this. This is a sodium-potassium pump, and in this example, here's our protein molecule that spans the lipid bilayer of the cell membrane, outside the cell here and inside here. And this one's called the sodium-potassium pump because sodium is going to be taken in, and it's going to change shape to hold on to the sodium and pump it outside of the cell, and it's going to release that sodium, and it's going to grab the potassium, change shape to hold on to them, and go inside the cell with the potassium. But in order to do that, we're going to require ATP to be broken up into a phosphate group and ADP, which gives off energy in the process. And we can just watch this as the slides go through. We see the sodium that's been given off outside the cell, and it changes shape to take potassium ions inside the cell. And you can look at this in the circular graphic that helps you see each one of those steps specifically. And again, that's the sodium-potassium pump. It goes against the concentration gradient, uses a carrier protein to move sodium ions and potassium ions against the gradient into and out of the cell. We're going to look at vesicle formation. Sometimes we have trouble getting macromolecules into and out of the cell membrane. So we're going to form vesicles instead to get them out of the cell membrane, and this is going to require energy, and it keeps the macromolecule contained for transport. So if we're talking about exocytosis, we're going to take and form a vesicle that's going to take something out of the cell, and endocytosis is going to form one that goes inside the cell instead. Let's look at these a little bit more specifically. Exocytosis is going to happen when a vesicle fuses with the plasma membrane, and this causes secretion to occur. The membrane of the vesicle becomes part of the plasma membrane, and cells of particular organs are specialized to produce and export molecules this way, such as pancreatic cells release insulin when the blood sugar rises too high. So this is a picture showing exocytosis, and we have the plasma membrane, and we have outside the cell here, inside the cell here, and this is a secretory vesicle. So this vesicle is going to merge with the plasma membrane, and then it's going to open up and it's going to release that. So the example that they just told us about was insulin needing to get out into the bloodstream, so these would be insulin molecules getting to the bloodstream. Endocytosis, on the other hand, is when cells take substances into the cell that they need. Now there's two different types. Phagocytosis is taking in large particles and particulate matter. Pinocytosis are liquids and small particles dissolved in liquid. I'm going to say something that's going to be some kind of gross, but maybe it will help you keep this in mind, the difference between endo and exocytosis. When you eat and drink, take things into your body, you take in particulate matter or solids, which is phagocytosis, or you take in liquids, which is pinocytosis, and they usually are separate. However, when you get rid of something such as you throw it up, you get it and take it out of your body by throwing it up, all this is mixed together, the liquids and the solids. I know it's gross, but exocytosis, there's just one type. You get rid of all of it at once, but endocytosis can either be taking in solids, which is phagocytosis, or liquids, which is pinocytosis. Receptor-mediated endocytosis is another method, a type of pinocytosis that involves what we call a coated pit. And we see the plasma membrane, and we have a vacuole. This is a paramecium that's taking this into the paramecium, and it surrounds it. We have solute forming a vesicle here, and this is a solid particle, this is more liquid. And this is the coated pit, and this is actually specific. You notice it has specific amounts of solute that is picking up and taking inside of the cell. So phagocytosis, pinocytosis, and receptor-mediated endocytosis using a coated pit. So this completes all of Chapter 4, and this is the second part of Section 4.2. We're going to do application projects in class that are going to help us understand these processes a little bit better. But the best thing that you can do is to take notes over each one of these sections. Make sure that you listen to the videos. Make sure that you ask questions in class and study for the test. Know your vocab. That's a whole new set of vocab, so make sure you take time to study that too.